wondered why you look like the way you do?
Why? Why does a banana look like the way it does?
Or why we don't look like dinosaurs or chimpanzees?
Or apes?
Today we have the answer to all these questions. We are going to be performing a DNA extraction by a very simple method which can be used both at home and in the lab.
Today I will be extracting DNA from strawberries using a method that can be utilized at home.
And I will be extracting DNA from a banana with a method which is used in the lab. The methods are almost similar, but the apparatus we use in the lab is slightly different than the apparatus we use at home.
But before that, Vinaj and Vicky are going to tell us what genetics is all about.
Hi, I'm Vinaj.
And I'm Vicky.
Today we're going to be talking about genetics. Genetics is basically the study of genes and the human DNA. It's a fairly new branch of science that comes in with the field of biotechnology.
Genetics is many applications in today's world, including crime, where it's used in DNA labs, as you've probably seen on TV. Theory of evolution, which you can study even more in depth with genetics.
So today we're going to be showing you a presentation using an already made video to basically explain to you what genetics is all about, giving the basic knowledge to you. Genes can be described as basic code of instructions that makes you. Genes are taken from your mother and your father and are then bound together to form you. All living creatures in the world have genes, and genes are the functional parts of DNA.
DNA is deoxyribonucleic acid. Chromosomes are consistent of DNA. All of our characteristics have a unit, and they're basically consistent of protein.
DNA is shaped like a ladder, twisting around its axis. This is called the double helix DNA, which exists in most evolved animals. Its rungs consist of 4 different chemicals called bases. These are adenine, thymine, guanine, and cytosine.
All the rungs of the ladder are composed of sugar and phosphate. Double helical structure of DNA was first formulated by two scientists, James Watson and Francis Crick. What are the 4 proteins of DNA determine our characteristics? Say our genes say if we become a banana and auspicious or a human. And our characteristics like if we have brown hair, blue eyes.
Chromatin fibers are located in the nucleus of the cell and appears a large stringy mass. When the cell is ready for division, they gather up to form chromosomes.
The chromatin fibers only are from the human gene, and the average human gene has 3.2 billion base pairs.
It would take the average person typing about 60 words per minute for 8 hours a day, 50 years to type out the entire human genome. That's how complex we are. If you unravel one molecule of DNA, it would be 6 feet long. If you unravel all the DNA inside our bodies, it would be long enough to go from the Earth to the Moon 6,000.
Start.
The human genome is like a giant hard drive, where all its information is stored in various stores. And if you take out the memory of the average human genome, you can store 175,000 HTML and 700 terabytes worth of information.
Out of our genetic code, 99.9% is the same as everyone else. It's that 0.1% that differs us. 2% of DNA is protein. The rest of it is known as junk. It's not that it doesn't perform any functions, it's just that we don't know what these functions are.
Before we start the experiment, you need to keep a few materials ready with you. For the home method, we need fruits, a small glass, common salt, washing detergent or hand wash, a larger glass, tap water, coffee paper, isopropyl alcohol, that is like rubbing alcohol or sanitizer, and a sieve.
The materials required in the lab are ethanol, funnel, beaker, washing detergent, measuring cylinder, spooling equipment, a petri dish, distilled water, and a filter paper. First, we are preparing the lysine solution.
For a lysine solution, we require about 30 ml of water. While doing this at home, you can take about 1/4 of a glass. This is roughly 30 ml of water.
In our lab, method, we use a measuring cylinder instead of a glass. We take 30 ml of distilled water in a measuring cylinder.
Then you take 1 teaspoon of common salt.
We're going to take 2 teaspoons of washing detergent. And mix it well. Now we have our lysine solution prepared.
For the home method, I am using strawberries to extract DNA. First, we take about 4 or 5 strawberries. I am using frozen strawberries, but at home you can also use fresh strawberries. Once you have sealed the Ziploc bag, mash the strawberries inside it.
You do the same in the lab method. You take a banana, cut it and put it inside a Ziploc bag and start the mushroom. After you're done mushing, we add the lysine solution which we prepared earlier to the mushroom.
Once it is immersed, you re-lock the Ziploc bag and mush it a little more, mixing it with the lysine solution. Once you have mushed it with the lysine solution, you should leave it for about 10 minutes. This is the mashed strawberries along with the lysine solution. The lysine solution helps in breaking the cell membrane and the nuclear membrane.
After you're done with this, the chromatin material comes in the solution. Now we take the solution and filter it. For me, using a filter paper and a flask.
While you're doing it at home, you can use a sieve and a glass. What we obtained are filtrates of banana and strawberry. Now we're going to be forming precipitates. For that, you take a small glass if you're doing it at home and pour the filtrate, the strawberry, into it about halfway.
I will be pouring my filtrate into a petri dish.
Following this, precipitation is the changing of a liquid state back to its solid state. For that, we add, for the whole method, we add isopropyl alcohol. Fill it about double of what it is currently. What we have now obtained is a precipitate.
Yep.
For the lab method, we use ethanol. I will add ethanol from the side to the petri dish side.
Having obtained the precipitate, we now have to put the chromatids from the DNA extraction of the strawberry into a mini tube. This is going to be left for cooling to obtain the chromatids.